Welcome to clickmyproject.com. Now we discuss about the product semi-automatic segmentation and classification of Pap seam cells. Now we discuss about the abstract. Cytological screening has widely used for detecting the cervical cancers. In this study, the semi-automatic PC-based cellular image analysis system was developed for segmenting classes, clear cytoplasmic contour and texture feature to support vector machine classifiers to classify the different type of cells. Two experiments were conducted to verify the classification performance. The cross-validation result of the first experiment showed that average accuracy of 97.6% and 98.3% respectively. The classification result gives the better result because of the SVM classifier. Though the experiment was conducted several times, it was more reliable and accurate. This proposed method provides a feasible and effective for the cell segmentation as well as the cell classification. The segmentation process along with the classification gives the more stability to the process. We evaluate the results of segmentation and classification which have provides the best result in medical science. Now we discuss about the flow diagram. At first the input image is taken from the dataset. Then the input image is undergoing for the pre-processing. In pre-processing we have to consider the major two things. First one is filtering, then second one is edge detection. In filtering we have to remove the noise from the image. To remove the noise from the image we use the median filter for the noise removal. After the noise removal the edge detection is carried out. For the edge detection we use the canny edge method for the edge detection. After the edge detection the segmentation is carried out. The segmentation is carried using the contour segmentation method and the segmented image is undergoing for the feature extraction. In the feature extraction we have to extract the local features such as area, length, width, perimeter of the image for the both cytoplasm and the nucleus of the image. Then the classification is undertaken. For the classification we have to consider the two things, train feature and test feature. The combination of both train feature as well as the test feature we have classify the image type, which type is the image. After the classification result will be in the text format. To achieve the better classification we use the SVM for the classification purpose. The SVM is abbreviated as an support vector machine. Now we discuss about the initial running procedure. First select the main file.m, then right click in that file, then select the third option, run. Now the initial guide window was opened. In that you can see the several buttons. These are known as the push buttons. These 6 are represented as an axis. Then one edit box is available below to the push buttons. Then click the button input image. Now the dataset folder will be open. Now several images are present in that folder. Select any one image from the dataset. After the selection of dataset image, the image will be shown in the axis 1 with the title original image. Then select the second button, pre-processing. After selecting the pre-processing button, the pre-processed image will be shown in the axis 2. The pre-processed image contains the noise filtering and grayscale conversion with the title pre-processed image. Then click the button cytoplasm. After clicking the button cytoplasm, the edges only the display in the axis 3, then the other background will be subtracted. The image will be shown as the title cytoplasm. Then click the button nucleus. After clicking the button nucleus, the nucleus only will be shown in the axis 4. The other cytoplasm and background will be eliminated. The edge of the nucleus is plotted in the axis 4 with the title nucleus. Now the cytoplasm and the edge of the nucleus will be separated. Then click the button segmentation. After clicking the button segmentation, the segmented result will be shown in the axis 6. The green color indicates the edge of the cytoplasm and the red color indicates the edge of the nucleus. The nucleus and the cytoplasm are separated from the background. After the segmentation part, we have to extract the feature from the image, so that we have clicked the button feature extraction. After clicking the button feature extraction, the feature which is extracted from the image will be shown in the UI table 1. These are the feature which was extracted from the image. The second UI table consists of the feature from the dataset images, that is known as the train feature. All the train features are located in the UI table 2. There are totally 20 train feature for the whole dataset because the dataset contains the 20 images. After the extraction of feature, we have to classify the image using test feature as well as the train feature. Now click the button classification. After the classification result will be shown in the edit box, and also the result will be shown in the command window. In command window also the each feature extracted from the image was shown. The local feature such as area, length, width, perimeter, which are calculated, which is shown in the command window, and the respected values are also shown in the command window. From these features extracted from the image, the test feature will be made. The class label will also shown in the command window. Now the class label is 1, so that the classification result will be shown as an normal.